This conference will now be recorded.
Hi, my name is Karen DeBose, and I'm Skagit County's Pollution Identification and Correction Coordinator. I'm here to talk with you today about our use of microbial source tracking, what worked and what didn't. I want to provide you with some things to think about if you're thinking about doing an MST project in your own area. For those who aren't used to the terminology, microbial source tracking, or MST, is often referred to as DNA testing. We're analyzing water samples looking for the DNA of bacteria that are specific to the gut microbiome of different types of animals. The theory is if we find DNA from a particular animal in a water sample, along with a lot of fecal bacteria, we can say that the fecal bacteria likely comes from that particular type of animal. With the help of the EPA lab in Manchester, Washington, we have been able to test for humans, cows, general ruminants, and dogs. This information helps us to decide whether we need to look for a livestock problem, a failing septic system, or poorly managed dog poop in someone's yard so that we can stop the pollution from happening. A key thing to understand at this stage is that MST only gives a reliable positive or negative result. It cannot tell us how much of the pollution is coming from each source. So there's two ways to go about this kind of study. These tests are pretty expensive, so we want to be conservative in our sampling. Our first approach in 2010 was to take a classic source ID approach and see if we could identify what sources were coming from each stream and how the markers changed as we moved down the river. We sampled the mouths of the major creeks along the main stem of the Samish River, and we did it on set sampling dates, so we were not targeting storms. The dots on this map show all of our sampling sites, which, as you can see, are pretty spread out over a 123 square mile watershed. At the time, again, we were able to look for ruminant and only ruminant and human markers, so no dogs or cows. The result was only presence absence, meaning that we can't say how much of each source is at each site, just that it is or is not there. Back in 2010, I think this was a very logical approach. We hadn't gotten very far in our source identification work, and we were looking for some big picture findings that would help us narrow down our approach. The results weren't quite what we'd hoped for. Humans and ruminants were found just about everywhere. Now that we've been working in these watersheds for a decade, that makes total sense. Humans and farms are scattered all over the watershed, so it's logical that we would find signs of them just about everywhere. At the time, it was really disappointing to not have data that could point us in a particular direction. What was really useful about the results was in answering some of our critics. The Cattlemen's Association particularly was very vocal that we were unjustly blaming the cows and that they couldn't possibly be a major source. With ruminant markers found everywhere, that was clearly not the case. We also found human markers everywhere, so we clearly had a lot of work to do. Fast forward 10 years or so, we found a lot of sources of bacteria from large and small farms, from septic systems, from piles of dog poop, and more. We've scoured the watershed over and over, but there are a few spots that we haven't been able to figure out. So we decided to try again with the help of the EPA Manchester laboratory. We paid sampling and shipping costs, and the EPA covered the cost of lab analysis. This time we targeted storms because that's when our pollution happens. We were able to test for a cow-specific marker, 2 human markers, and a dog marker, along with ruminants. And again, the results are presence absence, so I still can't tell how much might be coming from each source. I want to go over 2 types of areas that we looked at. Our first confusing area was located in Swede Creek. There are only 4 properties here, and we'd already visited 3 of them. We'd done 2 septic dye tests that came back clear. We'd improved livestock management on the property on the right, and we worked with the property owner on the left regarding dog poop management. But with all of that, bacteria levels weren't going down, and we were sampling the red dots, those sites. MST results showed 3 major things. Even though the septic system on the right had cleared a dye test, there was ample evidence of human markers. The dye test had been done in late spring when things were drying out, so we think that the septic system was seasonally failing due to high water tables rather than failed year-round. The other sites all showed evidence from humans, dogs, and ruminants, but not cows. So we needed to look closer at the RVs, the sheep, and the dogs that live in the 3 properties to the left. Thanks to these results, the sheep have been removed from the property and the septic system was fixed. The RV property is still in process. The next confusing area is in the Samish Flats near Samish Bay. There's lots of agriculture going on in this area with a dairy nearby and crop farms all over. And of course, some houses with septic systems and drainage ditches everywhere. You can see the red dots again are sampling sites. We see occasional high levels of bacteria in the rainy season, but the summer is when things get really interesting. There's no rainfall to push pollution into the ditches, yet we find hugely high levels of fecal coliform bacteria. And then sometimes we come back 2 weeks later and the levels are back near zero. Washington Department of Agriculture has been unable to find any smoking gun sources on the dairy itself. We had started to wonder whether the bacteria could be regrowing in the nutrient-laden runoff from those ag fields and causing those spikes. There's some indication in the literature that it's possible, and the literature also indicates that E. coli can also reside and grow in soil. After more than a century of dairy applications in the area, that would not be surprising. MST results showed some evidence of impact from cows, humans, and dogs during the rainy season. But during the summer season, we found no markers for cows, humans, ruminants, or dogs. So our source is either an animal that's not one of those, such as birds, or we have our first evidence for regrowth. And that's a really helpful answer because at least we've been able to say what the problem is not. There were more areas that we focused on, but this is a snapshot presentation, so I'll skip them. We learned several important lessons with this second round of MST. First, do your regular source ID until you've run out of options first. It takes more time, but MST is a pretty expensive technology, and if you want to move faster, you're going to pay for it with a lot of very pricey lab costs. And second, we finally got actionable results by focusing on relatively small areas where there were a small number of possible sources nearby. And with that, I appreciate you taking the time to view this presentation. I'm happy to chat more about the subject if you have questions. My contact information is here, and thank you.